All right. Chapter 17, Mechanisms of Evolution, Part B. This will be shorter than Part A. That was a little long. My bad, y'all. Okay, so when we're talking about evolution, sometimes we split it up into two categories. Sometimes you'll hear the term microevolution or macroevolution. So these are just phrases that help us to keep track at what scale of evolution we're talking about. So microevolution is small scale population level changes. And so here with microevolution, we might be talking about birds going from blue to red feathers, or from moths going from white to black, or black to white, or um, crayfish going from having a big right claw to a big left claw, or whatever. So microevolution is a lot of the stuff that we talked about in Part A. Macroevolution is something we'll talk more about in upcoming lectures, and macroevolution is more large-scale evolutionary changes. So I've talked a little bit about dinosaurs evolving into birds, so this would be an example of macroevolution. So like a dinosaur that has feathers, and then more feathers, and then feathers with wings, and then losing teeth, and so on. Those are big changes over a long period of time, and that would be macroevolution. And so basically macroevolution is microevolution just played out over a long time frame. So microevolution might be, a generation or a few millennia, but macroevolution, we're talking usually millions of years. So anyway, just keep those in mind. We'll come back to these a little bit later when we talk about different views on evolution. Also want to point out that evolution sometimes really just doesn't happen. In other words, sometimes a species will just remain more or less the same for really long periods of time. And when that occurs, this is called stasis. So in stasis, you get an organism where over time there may be some changes, but overall it's not really splitting off into new species, and it remains the same today as it has remained for perhaps millions of years. Two examples. One, the coelacanth fish, which you can see in the bottom slide. On the lower left-hand picture, here's a picture of a fossil of the coelacanth. And on the right is a photograph of a living coelacanth. So these things are still alive. And the time difference between when we see them as fossils and modern day is about 70 million years. So it hasn't really changed for 70 million years. The ginkgo tree is another example. So we found fossils of ginkgos going back millions of years, tens of millions of years, and it's still alive today. And the species that's alive today has remained virtually unchanged since it was fossilized back when the dinosaurs were around. So stasis is something that happens sometimes. Another thing that happens is coevolution. So I've talked a lot about one species evolving into two or whatever, but sometimes this doesn't happen just with a species by itself. Sometimes evolution happens in conjunction with another species. So coevolution is when two species evolve alongside each other. And as an example of that, look at the picture at the right. So you can see these flowers here. So these are a type of orchid. And this long green thing that hangs down, this is the nectar tube. So this flower is trying to attract pollinators to spread its pollen, and it's paying the pollinators with a sip of sugary nectar. And this nectar can be found here, way down here at the base of this nectar tube. So if you're a pollinator, how are you going to drink the nectar at the flower? Well, you need a really long mouth part. So you need a mouth part that would go into the flower and would be able to go way down this long nectar tube and would suck up the nectar from the bottom. And it turns out that this is the pollinator of the orchid. This is a orchid hawk moth, or Darwin's hawk moth. And this thing has a really long siphoning mouth part, just a long mouth. And you can see it here. This is a dead one, so they posed this probably in a museum. So here's its mouth part. So you can see it lands on the flower, and this mouth part goes way down and sucks the nectar. So how did that happen? Well, over time, this flower, basically, it's happy having one pollinator, this moth. And this moth is happy basically just drinking nectar from this one species of flower. So over time, this orchid got a longer nectar tube, and so that's evolution. And as the hawk moth drank nectar, the ones that had the longer mouth part were selected for. So over time, the hawk moth got a longer proboscis. And so over time, the flower would get a longer nectar tube and the moth would get a longer proboscis. And the flower would get a longer nectar tube and the moth would get a longer proboscis. So this is an example of coevolution. The flower and the moth both evolved alongside each other, and they were basically just, playing off of each other. I won't talk about these other pictures. So let's talk about some evidences of evolution. Again, this is straight from your textbook. So first is homologous structures. So homologous structures are body parts that have some similarities, and those similarities are thought to be because they both were passed along from a common ancestor. So in the picture on the left, you can see we've got a human arm, a cat leg, a whale flipper, and a bat wing. And where this is an arm and a leg and a flipper and a wing do very different things. But at the same time, if you look at the bone structure, you see they're really, really similar. So we've got, in the human arm, we've got one bone, two bones, and then a bunch of wrist and hand bones. In a cat leg, it's one bone, two bones, a bunch of ankle and toe bones. And a whale flipper, we got one bone, two bones, and then a bunch of smaller bones. And in a bat wing, it's the same. So why do all four of these have the same bone pattern of one, two, and then lots of little bones and then long fingers? Well, the thought is it's because they're homologous. In other words, that bone structure was inherited from a common ancestor. So sure, over time, humans, cats, whales, and bats have all modified this basic blueprint, this basic bone structure, into arms and legs and flippers and wings, right? Which are very different appendages. But the point is that basic bone structure came from a common ancestor. And we can zoom out and look at an even larger scale. So here's a reptile that we know from fossils, and it's got one bone, two bones, little bones, and then finger or toe bones. And if you compare all these different limbs from a pterosaur, a chicken, a penguin, a porpoise, and so on, you see that same pattern: one bone, two bones, and then smaller bones. So it's modified into different types of wings, wings that are flippers, limbs that are flippers, arms that are wings, arms, and then this is an elephant leg. And so again, all of this basic pattern evolved from this common ancestor. They're all playing with this original design. How do complex things arise? Well, it's a lot more complicated and difficult to explain, but this is this is ongoing in biology. In other words, biologists attempt to apply the principles of evolution to understand how complex structures evolve. So let's talk about the human eye, or a complex camera type eye. Actually, it's not humans, but in the picture we've got a squid. So a squid eye is really complex, and it's good for seeing stuff. It's an eye that functions really well. And it's got a retina here, and this absorbs photons and then transduces them to nervous impulses and sends a signal to the brain through the optic nerve. And then the brain creates an image. There's a lens that focuses light. There's a cornea that helps protect the lens. And so that's a complicated eye. How did this evolve? For a long time, people and scientists said, how in the world did a complex eye evolve? Because it would take multiple steps. So to explain that, we can start with the most complex and let's start with the most simple and work our way forward. So in each case, these different eyes represented on this picture are simpler eyes that exist on some organism that we can find on Earth. So here's the eye of a limpet, which is like a little barnacle. And you can see that it's got cells, and some of these cells that are on the outside of its body are light sensitive. So light hits these cells, transduces them into nervous signals, which sends a signal along a nerve to the brain. so it's able to see light. Now this eye is really terrible. Really, they can just detect where light is. So this limpet can't really, see anything. I mean, it can see, but it can't watch Netflix. All right, it's not a very good eye. This is the eye. Oh, man, where is it? Here's an eye on a type of mollusk, and you can see that basically it expands this simple eye, and all it does is fold it. So the light sensitive cells are now kind of protected in a little cup. And so simply by bending these cells into a U-shaped pattern allows this organism to tell where the light is coming from. So this eye, you still wouldn't be watching Netflix, but at least you know where the TV is. This is in a nautilus. So this is a little shelled aquatic critter, and it has this eye cup, but it's got a fluid-filled cavity. So in other words, this little empty space here is filled with fluid. So again, light's going to come in, it's going to get distilled, or it's going to get filtered through the fluid here, and then it's going to hit the light sensitive cells, and this nautilus will be able to see. And so it will be able to kind of see. It's not going to be able to read the closed captioning on Netflix, but it can maybe tell what's going on. The fluid-filled cavity is going to help it focus light. Here's a marine snail. So it's got this general U-shaped pattern of light-sensitive cells. It's got the fluid-filled cavity, but then it has a lens, which is even better at focusing light, and then it's got this thin, transparent tissue on top that protects it. So this is more complicated than that, but not quite as complicated as the squid eye. So the squid eye just takes this design, and this tissue becomes the cornea, and it's able to just better control light coming in. It's going to be able to, this squid could sit down and watch Netflix with you. It could tell what's going on, read some closed captioning if it was written in squid language. So we can see just in existing organisms, a simple eye to something in between to a more complex eye. And so it's thought that these eyes that we see in nature are just representing possible intermediate steps in the evolution of a complex eye. So in other words, the first organism to evolve an eye had an eye something like this. And then in some lineages over time, it got improved to this, like in these mollusks. And then some lineages, like with the nautilus, they got improved to this. And then in the marine snails, it got improved to this, and so on and so forth. So complex structures, it takes a lot of work to try to figure out how they evolved. But this is an example of one way that that biologists will explain the evolution of a complex structure like an advanced camera type eye. Let's see. Okay, so in evolution, we can also look for evidence of evolution by examining genetic information. Right, so we can look at DNA. We can compare the A, C's, T's, and G's of one organism to another. We could also compare the RNA sequences. So we can look at the messenger RNA or ribosomal RNA. We can also look at the amino acid sequences. That's what's going on down here. Each of these little blocks is a different amino acid that's coded for by the DNA. So in other words, we can use DNA at its multiple functioning levels to compare between species. And the idea is, if two species shared a recent common ancestor, then we should expect a lot of overlap. And the more distant the common ancestor was, we would expect some overlap, but we would expect it to be, we would expect them to be less similar. So as an example, instead of DNA, let's think about a term paper. So let's say someone wrote a paper about how awesome biology is. And then the next year, they passed that paper on to the next generation of students. And some of those students took that paper and basically just changed a couple of words and turned it in, right? And the next year they passed on those copies of the term paper, and the next generation of students changed a few words and phrases a little bit. So we could compare, and we could see that the third generation term papers are fairly similar to each other, but they're more similar to generation 2 than they are to generation 1. I hope that made sense. So we do the same thing with genes. And in down here, what we've got going on is we've got 3 amino acid sequences. This one's a yeast, which is a type of fungus. This one is a wheat, which is a plant, and this one's a primate. So this would be, I don't remember if this was human or chimpanzee or what. And you can see that the amino acid sequences have sections for this particular protein that are the same. So this sequence is the same, this one's the same, and this one, well, isn't. Oh, I'm sorry. Man, I got that backwards. This is different from this one, which is different from this one. But this amino acid is preserved. In other words, it's the same across all three. So you can see that there's big chunks that are the same across the three species, but in some cases, there's single amino acids that are different. You can see that, anyway, the point is, we can use these sequences to kind of compare similarities and kind of get an idea of how closely related different species are. So to expand on that idea, let's look at mouse and human chromosomes. So let's look on the right. So these are the chromosomes that we find in human beings. So here's the 22 autosomes. Each one is color coded, right? So autosome number 10 is color coded. Shoot, what color is that? What is that? Purple? Let's go with purple, right? Here's the sex chromosomes, X and a Y. So this is from a male. And so they're just color coded, right? When you look at mouse chromosomes, so first off, mice have a different number of chromosomes. So they have 19 autosomes, and then they have 2 sex chromosomes. And if you look at their chromosomes, it's kind of like a jumbled mess of human. So human chromosome 1 is dark green, and we find it kind of spread out over different mouse chromosomes. So some of the sequences on the human chromosome 1 we find over here on mouse chromosome 1. So this section would overlap with this one. We find some human chromosome 1 over here that's on mouse chromosome 8. And we find some human chromosome 1 that's on mouse chromosome 4. So these sequences would be really similar to human chromosome 1. They're just in different places. They're spread out over different chromosomes. So this means that humans share a lot of the same genes as mice, but they're arranged in different ways. So going back to the term paper, let's say term paper generation number 3 was, if we were to compare it with generation 1, we might find that lots of the sentences are really similar, but in the first term paper, maybe the first paragraph is in the first paragraph, and in generation 3 term paper, the paragraph that was paragraph 1 is now paragraph 3. So in other words, we look for not just similarities in the DNA, but we also look for how maybe they've gotten moved around over time. I hope that made sense. If not, see me on the discussion board. I'm happy to revisit this. What about humans? Humans have a lot of similarities with other primates, including chimpanzees. So if we say that humans share 100% of the same genes, even if they're different alleles, it's the same genes as other humans. When we compare the human genome to chimpanzees, depending on how you calculate it, it's 97 to 99, let's just say 98% identical. If you line up the human and the chimpanzee chromosomes, here's human chromosome number 1 next to chimpanzee chromosome number 1, human chromosome number 3 and chimpanzee chromosome number 3 lined up next to each other. I've skipped 2. We'll come back to that later. And these are color coordinated. So on human chromosome 3, it's got some genes or a gene that's color coded red, and you see that on the chimpanzee chromosome, the gene is really similar, which is why it's the same color, and it's also in the same place. So if you just kind of eyeball this, you see that humans and chimpanzees are pretty similar. They have very similar DNA sequences. So in the term paper, the sentences would be very similar to each other. But also, those genes are in more or less the same places. So where paragraph 1 of term paper number 1 is also paragraph number 1 of the 3rd generation term paper. So again, we're able to use DNA to look and compare how similar different species are. And the more similar they are, the more recent they shared a common ancestor. So if you compare the human DNA to a New World monkey, a monkey that's in South America, it's about 84% similar So we shared a common ancestor with New World monkeys a lot longer. It's a much longer time frame than when we shared a common ancestor with chimpanzees. So just get a sense of scale. This is about 7 million years, and this is probably closer to, I don't know, 10, 11, 12 million years. All right, going back to chromosome 2. Here, actually, let me return to this slide. So human chromosome 2 looks like this. And on chimpanzees, they have 2 chromosomes where one chromosome looks a lot like the top portion of human chromosome 1. And they have another chromosome that looks a lot like the top portion of human chromosome 2. Okay, so what's up with this? So we really have 2 options. It could be that the common ancestor had 1 chromosome, and then in the descendants that became chimpanzees, it split into 2. Or the common ancestor had 2 chromosomes, and in the lineage that led to humans, it fused into 1. Well, when we look at these other apes, it turns out that they have 2 chromosomes as well. So the leading idea is that, or the best explanation is that all these, so somewhere in here the common ancestor had 2 chromosomes, and then somewhere in here, in the human lineage, those 2 chromosomes fused into 1. So that's called chromosome 2 fusion. So you can see it here. Gorillas and orangutans and chimpanzees, they have 2 chromosomes. But in the human lineage, they fused into 1. We're just going to ignore inversion for now. Right here, you can see it. In orangutans and gorillas, it's 2 chromosomes. All right, and in chimpanzees and humans, these 2 chromosomes have fused into 1. So it's not just the DNA sequences that are used as evidence to support evolution. It's also the order of the genes and where we find them on chromosomes. We also use or look at pseudogenes to try to figure out evolutionary relationships. So pseudo means fake and gene means gene. That's cool. So a pseudogene is a fake gene. All right, and this, what that means is it's just a gene that doesn't work anymore. So it might be surprising to learn that a lot of the genes in your genome are just broken. So there's DNA there, but they don't really code for functional proteins. Sorry, I lost my train of thought. What was I saying? So let's look at this. This is the gulo gene. So don't get too freaked out. It's just a gene, and biologists give them names. So this gene is called gulo. All right, and gulo codes for proteins that allow an organism to make vitamin C. In humans and apes, the gulo gene is broken. And since the gene is broken, it doesn't make a functioning protein, doesn't make a protein at all. And so in humans and apes, we can't make the protein that helps us manufacture vitamin C. So therefore, in apes and in humans, since we can't make vitamin C, since the gulo gene is broken, it means we have to get vitamin C from our diet. So this is why you took, Flintstone pills or gummies or whatever you guys take as a kid, or maybe you still take vitamins. You should take vitamins. If you don't take vitamins, do it. So why in the world would humans have a broken gulo gene and chimpanzees and orangutans have a broken gulo gene, but at the same time other mammals like rats, dogs, and cows, they all have a functioning gulo gene? So in other words, a dog has a functioning gulo gene, so you don't have to give your dog vitamin C. You don't have to get it. It might need other vitamins, but it doesn't need vitamin C. So why is that? Well, an explanation is that in the common ancestor that led to orangutans, chimpanzees, and humans, the gulo gene got a mutation that caused it to stop working. And that broken version, that pseudogene, got passed on to orangutans, chimps, and humans. Why did natural selection select against this mutation? In other words, if the gene is broken, then should natural selection remove it from the gene pool? Well, it should if the broken gene is going to cause problems for the survival and reproduction of that organism. But it turns out in primates, having a broken gulo gene is fine. So if we can't make our vitamin C, which is bad, but primates eat a lot of fruit and they eat food that's high in vitamin C. So an orangutan having a broken pseudogene or having a broken gulo gene doesn't really affect its survivorship because it can get vitamin C from its diet. All right, so we can use pseudogenes like this in order to try to figure out evolutionary relationships. Here's another example. Well, actually, same example. Here's what a functional gulo gene looks like. So these black boxes represent sequences. So it's not just that humans, chimpanzees, and other primates have a broken gene. It's broken in the same way. So in all primates, this are all mutated, whereas this section is not. It's intact. This is mutated, this is mutated, this is intact, and so on. And so it's not just that we all inherited a broken gulo gene, it's we all inherited the same type of broken gene. All right, so again, that's used as evidence to say that primates share a common ancestor, and that these primates are all closely related. And while we're similar to a rat is a more distant relative because it has a functioning gulo gene. Here's another pseudogene. It's for vitellogenin, and vitellogenin is a gene that codes for egg yolk. All right, and so in animals that lay an egg, especially mammals that lay an egg, or it doesn't have to be mammals. In animals that lay eggs, their vitellogenin gene codes for egg yolk. Well, in humans and in placental mammals, so in chimpanzees and pigs and elephants and so on, we don't need egg yolk because we have a placenta. So in placental mammals, the vitellogenin gene broke somewhere around when we became placental mammals, and so we've all inherited a broken version, right? And so in humans, the vitellogenin gene, it's got this broken section. It looks like this, and it's in between a gene we'll call A and a gene we'll call B. All right, so in humans and all these other placental mammals, the vitellogenin gene is broken here, and it's in between gene A and gene B. If we look at a chicken, we find that it has a functioning vitellogenin gene. Now, that shouldn't be a surprise because they need this gene to work because they make egg yolk because they're laying eggs. But we also find that their vitellogenin gene is in the same place as our broken version. So it's in between gene A and gene B. So again, chickens are thought to have inherited gene A, the vitellogenin gene, and gene B, whereas placental mammals inherited those three, but the vitellogenin gene got broken somewhere along the way. So again, lots of different ways that scientists look for ancestry and similarities between humans under the paradigm of evolution. All right, that's part B.